So good morning, everyone, and welcome back to day 2 of the ILAR Roundtable on Reproducibility. My name is Pam Chamberlain, and it is my honor and privilege to be the moderator for this morning's session. And my hope is that day 2 will be every bit as much informative and engaging as day 1 was, and since I'm moderating the session this morning, I'm hoping that it's going to be even better. So help me with that, okay? And it's my job to introduce the speakers and keep everybody on time, and I take that responsibility very seriously, and I'll appreciate cooperation from everyone to help me do that. Thank you. So the first session this morning is on reproducibility challenges in the future of animal models. And without any ado, we'll get started and introduce the first speaker, who is Dr. Roger Reeves, who is a professor in the Department of Physiology at Johns Hopkins University School of Medicine. Please welcome Dr. Roger Reeves.
Thank you very much. I'm very glad to be able to join this meeting. I'm sorry I couldn't be here yesterday. Life has been happening to me at a rather rapid pace the last few days. So what I want to tell you about today, of course, are issues that have arisen around reproducibility of studies in mice that appear in the literature. And to sort of paraphrase the charge I was given, what are the new technologies that are coming out in mouse now that really are transformative for how we look at genetic models in mice? And will these allow us to make new advances to save us from these problems of reproducibility? And I put the second part in there because it's the only clear answer I'll give you today, and the answer is clearly no. Technology is not going to save us. Oh, okay, for the webcast. Can the rest of you also hear me? Yes. Excellent. Okay, so technology won't save us, and in large part I think that's because technology is not really the root problem. As in so many things, it's people who are the root problem. And in science in particular, this is an endeavor undertaken by people who tend to be fairly compulsive, fairly hardworking, fairly intelligent, and the people who really exceed and push the edges of fundamental knowledge tend to be unusual people and very, very capable people, although albeit maybe in a very small area. And the fact of the matter is some people are better at doing some things than other people are. So if you get discoveries that are reported and you hear, oh, 90% of the things in nature couldn't be replicated, the truth is the people who are trying to replicate those are usually not the people who were able to get the experiments to work in the first place. And I think that's kind of an issue. The second thing, so I'm getting all my gripes out of the way up front. So the second thing that I think we have a real problem with is an understanding of what the scientific literature actually is supposed to be, and that's certainly misunderstood by the public, by the popular press, and I think also by scientists to a degree that's quite concerning. It seems that we've come to a point where anything that's published in a scientific journal is supposed to be absolute, infallible, incontrovertible truth. And that's just not what the literature is. It is, in fact, our stock exchange of ideas, just as anyone can form a company and take it to Wall Street and see with the collective wisdom, impressions, whatever it may be, the collective gestalt of the markets, is this a really good and viable idea or not? That's what the literature is supposed to be for scientific discoveries. It's not a claim that this is absolutely true and there can't be any deviation from it unless there was fraud or corruption or some crime like that. So to take away that sounding board for the community is a really serious concern in my regard. And sort of an extension of that is there's little reward in the structure of doing science today in an era of really reduced budgets, but a greatly expanded scientific enterprise. Little reward for doing science properly, which of course is to form a hypothesis, do everything you possibly can to disprove that hypothesis. When you can't do that, submit it for publication so 3 or 4 other people can try and disprove it and come back with some ideas for you to improve it. And when they can't disprove it either, you publish it, and then the entire world this stock market of ideas, if you will, can work on that idea and say, you know, there's a lot to this, but here's a part over here you didn't think about correctly, and that's where we need to move forward. And I think that's been replaced with a substantial reward in terms of grants, promotion, what have you, of the desire to get a paper, which makes me absolutely crazy. But now I'm done venting, and I will reassure Dr. Chamberlain I'll be back on path here for what are we going to do with mice in this particular regard. So the first place to start, I think, is why do we use mouse models in the first place? And the fundamental reason for this is that there are genetic structures in mice that are not found in nature. And specifically, I'm referring to the ability to inbreed mice essentially to homozygosity. There is no homozygous mammal in the world. Mice can survive that way when they live 4 inches from all the water they need, right on top of all the food they need, and their cages are changed all the time and so forth. But these are not natural structures. And that provides a lot of genetic power, but it also comes with some costs that I'll discuss a little further. Mice are useful because you can make your own genotype. If you don't like the way the chromosomes are arranged or the way this gene looks, or you want a human equivalent of that gene over there, you can do that in mice. I'm not going to talk more about making your own phenotype today, but very large screens can be done in mammals with mutagens and so forth, if you have a particular phenotype of interest. And all of this is relevant to us because comparative genetics allows us to compare this back to the human genome. So the inbred mice, I think a real misunderstanding among many people who don't use mouse models a lot is that somehow all inbred mice are identical in terms of their phenotype. That's absolutely not true. The reason we have a complex genome, the reason that we could not readily reconstitute the human species by taking any appropriately sex-matched pair from this room is because there is not sufficient genetic variation in 2 individuals to buffer the species against all the effectively infinite variation in the environment that they have to be buffered against. So, in fact, there are some very concrete examples of one example of development of the skull that is much more highly variable between individuals of an inbred strain than it is in an outbred strain, and that's presumed to be a trait that is buffered by genetic variation. And when you lose all that buffering by going to homogeneity, you actually increase variation. So I think that this is an important thing to keep in mind, and it's really a matter of education. As with anything, any technology you use, any system you use, you have to understand it. You can't just kind of buy it off the shelf and think that's going to work. So I've been so bold as to make 2 recommendations in the course of the talk. The first one is, I think in writing grants and in publishing papers, one should justify the use of inbred versus outbred mice. Fairly simple, I think fairly clear recommendation, but especially if you're trying to model a condition that occurs in human beings, you should justify the use of an inbred mouse because human beings are outbred. Simple statement. The collaborative cross, I'm getting wordy here, is just a very interesting experiment in producing a lot of variation, but with some control and knowledge of the origins of this. This started out to produce a lot of recombinant inbred strains, new mouse strains that are inbred, but are a combination of 2 established strains. But this was actually done with 8 different strains of mice representing 3 different species of mus. So it captures a huge portion of the variation of the genus, actually. And there have been some very interesting results, especially from Gary Churchill's group at Jackson Labs, about the intermediates here in diversity outcross, they call it. Okay, make your own genotype. This is at the heart of the technology question. Of course, for decades now, we've been able to make transgenic mice to introduce new DNA that integrates into the genome of a mouse, carrying various kinds of markers with that. The development of embryonic stem cells allowed us to knock out, make null alleles, which also occur very, very rarely in human disease, and knock-in, which are now more popular, places where we replace a normal coding sequence, wild-type coding sequence, with a mutant sequence. The new synthesis is genome editing, shorthand CRISPR, which I'm going to tell you a little bit more about because I really think this is a transformative technology in terms of being able to edit the genome at a very fine level. And chromosome engineering, if you don't like the way chromosomes are configured in mice, you can actually just remake them. This has been very valuable in my own research in Down syndrome. Chromosome 22q deletion syndrome has benefited from this, and a number of other conditions as well. Okay, so if you want to make your own genotype, the first place you go is not the laboratory, it is your computer. The Knockout Mouse Project made null alleles for thousands of genes and reached their goals, and that has now morphed into the International Mouse Phenotyping Consortium. And in the IMPC database, there are more than 36,000 genes that are either assigned for targeting. These are largely conditional alleles where you can decide what tissues and what time you want to delete a gene. There are ES cells available, there are mice made, and/or there are mice that have been phenotyped. Phenotyped, I think, is another Achilles heel here, and I'm going to come back to that when I get done explaining this. Okay, so just to remind you, to make a transgenic mouse, what we do is we take a very, very small glass needle, put it into a solution of DNA, isolate zygotes from mice. In mice, the pronuclei do not fuse. They just, the nuclear membranes break down at first mitosis, or first cleavage. And inject 2 or 3 picoliters of the DNA-containing liquid directly into the pronucleus. And at some reasonable frequency, if you did everything well, that DNA will integrate in random sites in the mouse genome. And that's just a picture of that. If you're really good, that afternoon you can put them back into the mouse, or if you want to be a little more cautious, you wait overnight to make sure that the zygotes actually cleave and you didn't kill them in the process of sticking a big needle in there and blowing up a pronucleus. And then you just put those back into the uterus with a 26-gauge needle, and about 3 weeks later you get a bunch of mouse pups. And a week after that, you can make DNA from those pups and type them to see if they have an integration of your transgene or not. So it's a pretty rapid process once you have your construct in hand. These can be used to model dominant genetic conditions. If you have a dominant mutation in a gene, you can simply inject that dominant mutation, and presumably it will show its effect immediately in the offspring. You can create genetically marked populations of cells. You can survey genome regions by making a construct that has what you think is a promoter in it or an enhancer, and divide this up as long as you have time and patience and money to do so. And it's actually the transgenic technology that is the basis for CRISPR gene editing. So the fact that this is pretty rapid and can be done efficiently and then enhancing that efficiency many dozen-fold with CRISPR is what's important about this. Embryonic stem cells, or on this thing that I grabbed from nature here, induced pluripotent stem cells, produce a cell that can be manipulated in the tissue culture laboratory to change the genome in ways we want, usually based on homologous recombination. And using these cells then, we can edit the genome in the laboratory and then put these back into mice by a slightly different route. Here we use an older embryo. This is a, pardon me, a blastocyst staged embryo with the blastocele cavity here, an inner cell mass over here. This is a considerably larger needle than we use to inject a pronucleus, and it's full of embryonic stem cells that can be squirted right into the inner cell mass here. And in some percentage of time, depending on how carefully you've raised those cells, they will incorporate into the embryo. The outer cells here, the trophectoderm, are going to contribute to the fetal part of the placenta. The inner cell mass is what's going to become the organism. And so these cells are capable of becoming part of that and then contributing to every tissue in the body. And that, of course, includes the germline. So you get out of these chimeric mice, mice that had 4 parents, although each cell only had 2 parents. But in this case, in the case of these mice, one parent was dark pigmented and one was light pigmented. And these animals, as I say, some percentage of the time will have incorporated the embryonic stem cells into the germline. So you can then breed those, look for germline transmission. Once you get that, then you can expand your colony and do the experiments. I've mentioned a few more steps than I did in the first one, and in fact there are quite a lot more steps. This is a time-consuming and pretty labor-intensive and relatively expensive process to go through. But it gives you much more control than standard transgenesis does. So you can model recessive conditions here by replacing a normal gene with a mutant gene. You can create genetically marked populations of cells again and find expressed genes, and express genes conditionally. So you can set this up so you can express a gene at a specific time in a specific place in the mouse. It's very powerful that way. And I've been dropped. Houston, we have a problem. Okay, thank you. Okay, so the newest twist on this is genome editing. This is done using nucleases, enzymes that cut DNA, and there have been really 3 iterations of this. Zinc fingers and talons both are quite powerful, but also pretty complicated to use and pretty expensive. The CRISPR has made the revolution in the last year, really, coming into the public domain to really open up what one can do in terms of genome editing. The CRISPR system has 2 or 3 elements. It has an enzyme, which is shown in purple here, called Cas9. This is, you can think of this as a bacterial immune system enzyme. It attacks foreign DNA and cuts it. The second element is a guide RNA, which associates with a complementary sequence in genomic DNA and tells the enzyme where to cut. And if you want to do something like make a single nucleotide replacement, when you cut the DNA at a target, you can also include a small oligonucleotide, which, for example, would have a single base pair change that changes one codon and gives you a point mutation in the resulting thing. So genome editing has a lot of advantages. You can do your genetic engineering in vivo instead of spending a year in cell culture doing that. The frequency is vastly higher than the old method of doing this in stem cells, although you can use CRISPR in stem cells as well if you want to increase that frequency, which, why wouldn't you want to increase that frequency? It can be used in anything that has DNA and is alive. So anything from bacteria, invertebrates, vertebrates, plants, you can do all these. In mice, you can often target both alleles in one generation and get homozygously targeted mice, and you can target multiple loci in one generation. So you can put in 3, 4, even 5 constructs, and some percentage of the time hit all of those. One of the biggest disadvantages is off-target cutting, but there are now computer algorithms to predict where those sites will be, so you can easily screen for those and breed them out. And sometimes it's valuable to actually have a cell where you can test your construct if it's a complex regulation ahead of time. This is why CRISPR is so much better. This is a rather abbreviated and optimistic look at what it takes to make a mouse using standard homologous recombination in embryonic stem cells, and a conservative estimate is 1 year if everything works perfectly. The cost of the construct, $3,500 in our core to make the ES targeting, $4,850 to make the mouse. You need somebody who's fairly talented, spending probably 30% of their time on this for a year. This process not infrequently goes to 2 years, so it's quite expensive and quite long. CRISPR, the constructs are pretty easy to make. A month is not an unreasonable amount. The pronuclear injection, you inject it, takes 3 weeks to make the mice, another week to let them grow up so you can screen the DNA, usually by PCR and sequencing. If it's taking your student longer than 1 week, you need a new student. And then you can expand your colony, breeding out the off-target effects in that. So it's really very, very rapid. The cost is a third to a quarter to maybe even less than that. So I think this is going to lead to an increased use of mouse models because they're going to be accessible to many more investigators. So my recommendation number 2, it has to do with phenotyping those mice. I think this is a big problem. And the fundamental shorthand for this is that not all tests that have the same name are actually the same test. So a couple of years ago, a student and I surveyed the literature for all the behavior tests that had been done in mouse models of Down syndrome, the area we work in. We found numerous citations of tests. In one in particular, which is called novel object recognition, we found 11 different papers that cited it. No 2 of them had used the same method for the test that they gave the same name to, and this included the same lab which had done it multiple times. Every time it was different. So I think some kind of standards for especially behavior, pharmacology, metabolism. Who should set these standards? There's a loaded question. How standardized should it be? I would say a very minimal core that could be used to relate experiments. You don't want to stop the creativity of the process to phenotype correctly, but you do need some way to say, is this really on track with what we've known previously? And then, so that's this directive here, and I believe that's it, and I almost made it on time. I think we'll have discussion in a minute, is that right? Yes. Thank you.
Thank you, Dr. Reeves. Our next speaker is Dr. Jeffrey Rogers. And Dr. Rogers is Associate Professor in the Human Genome Sequencing Center and Department of Molecular and Human Genetics at Baylor College of Medicine in Houston, Texas. Thank you.
Well, I want to thank the organizing committee for the invitation to participate. Yesterday was a very interesting day, and I think today is going to be just as interesting. So thanks very much for the invitation for me to be here. Let's see. Yesterday we talked quite a lot about the broad problems of irreproducibility in animal research, and sort of there are issues that are general to all of those kinds of systems and all of those kinds of circumstances. I'm not going to talk about the general problems, but I thought my mission today is to talk about non-human primate work specifically. So what I'm going to do is focus on issues related to reproducibility in non-human primates. But first I want to remind you that non-human primate research is extremely important because primates share very close physiological and genetic similarity with humans. If work can be done and questions could be answered in non-primate models, we certainly should do that. But there are a large number of questions where the modeling simply can't be done in a non-primate model, and we absolutely need to do work in non-human primates. Some of the examples of that are infectious diseases, various aspects of neurobiology, and various aspects of endocrinology, reproductive biology, etc. So when we need a model which accurately simulates the human situation, very often the very best model, in some cases the only model, is going to be a non-human primate. So the goals of primate research in biomedicine are really to use non-human primates to provide unique information that cannot be derived from other species about human diseases and their prevention or their treatment. In addition, primates are often used for understanding fundamental aspects of human biology outside of the issues of health and disease. And finally, although it's not really our topic for this meeting, there's a certain amount of primate research that goes on just in terms of basic comparative biology and primate conservation. So what are the factors that influence reproducibility in primate research? One of them that I think is very important, and Roger actually just alluded to this in mouse studies, is differences in methods and differences in protocols across laboratories. But that's not really specific to primates. That's general across all of animal research, so I'm not really going to talk about that. What I do want to focus on for just a few minutes is small sample sizes, because this is a problem which, although it's general, I think it's even a bigger problem in primates than it is in other species. What are the drivers that push researchers to use very small sample sizes in primate research? Well, one is just access to animals. It can be difficult to get access to primates, but there are primate research centers that provide significant access, and really although that's a small issue, it's not a major issue. I think the major issue driving small sample sizes in primate research is that primate research is always a bit more expensive than non-primate research. Now, that has implications in a couple of different ways. First, whenever something is expensive, people tend to put a little more thought and care and planning into the execution of that study. Now, I'm certainly not going to try to convince you that every non-human primate research study is exquisitely designed and absolutely carefully planned. But I think that when things are expensive, people do put more planning and thought into it, and so I think that there may be a benefit there to the expense. However, the cost is really driving people toward very small sample sizes, and that's true for macaque research, for baboon research, for all primate research. But I want to give you just a little bit of an example from my own experience. Before coming to Baylor College of Medicine, I was at another institution where I was on the IACUC, and in fact, for a number of years, I was chairman of that IACUC. And in that institution, we managed macaques and baboons, but we also managed chimpanzees. So the IACUC on which I served handled a number of different protocol requests for studying chimpanzees. Now, if macaque research is expensive, chimpanzee research is even more expensive. So the cost factors are truly important when it comes to chimp research. And as a result, we had a number of cases where biotech companies, small biotech firms, even academic researchers proposed to our IACUC studies of chimpanzees with a sample size of 1. And when I was a member or the chairman of the IACUC, I always insisted that whenever we had a protocol proposed to us that had a sample size of 1, that we thought very carefully about the implications of that, because obviously there are any number of problems with reproducibility when you have studies with a sample size of 1. But in fact, we did approve studies with sample size of 1 because the investigator was able to convince us that there was a rational reason and a scientific justification. But I think I use that just as an example to illustrate that when costs are high, sample sizes and issues of sample size become very important. So now I want to switch, and for most of the rest of my talk, I want to talk about another factor which is very important in reproducibility in primate research, and that is genetic differences among study subjects. As Roger just gave a wonderful introduction to my talk about outbred species, the mouse models are a very different situation. Primates have significant amounts of genetic variation, and in fact, I'm going to talk about variation in 3 different levels. First, differences among species, although I'm not going to talk a lot about that. Then differences among populations within a given species, and then differences among individuals within a population. So each of these different primates is used in biomedical research, and each has its own particular strengths and weaknesses. It's questions where it's appropriate and where it's not appropriate. I certainly don't have to demonstrate to this audience that different species are going to have different strengths and weaknesses. But the dominant species in biomedical research are these 2, the Rhesus macaque and the cynomolgus macaque. The other species listed here are also important, very important, and widely used, but these 2 are really the most important species. I just want to briefly point out that the choice of species in biomedical research is actually very important, and once again, I probably don't need to belabor this to this audience, but I just want to briefly point out a couple of quick examples. First, if you're interested in prostate biology, prostate cancer, maybe you're interested in... Thank you. Maybe you're interested in prostate-specific antigen and its predictive value for prostate cancer. It makes a difference whether you choose an old-world monkey like a macaque or a baboon or a New World monkey like a marmoset, because the gene for calcrene-3, the gene that codes for prostate-specific antigen, is present in macaques, baboons, apes, and humans, but the gene simply isn't present in other more distantly related species. Some species, like baboons, are continuous breeders, so for reproductive biology and reproductive experiments, baboons are going to have a different kind of reproductive biology than seasonal breeders like Rhesus macaques. And one example that I think is very telling is marmosets. Marmosets are a really valuable primate. They're small, they're easy to handle, they're wonderful for a number of different applications. But marmosets have a very unusual reproductive biology. They almost always twin, and they produce dizygotic twins. But they don't produce dizygotic twins like humans do. In fact, the dizygotic twins share a placenta, and they share a structure of their placental circulation, so that hematopoietic stem cells that begin to develop in one of the co-twins actually break free, enter the placental circulation, and can populate the bone marrow of the co-twin, which is their sibling, but not their identical sibling. So if you go to adult marmosets and draw a blood sample from an adult marmoset, anywhere from 10 to 50% of the white blood cells that you collect in that sample will not actually come from the germline of the animal that you bled, but actually are derived from the germline of its co-twin. And remarkable or not, I have actually met researchers who were using marmosets for various kinds of research and weren't aware of that particular fact. Now, that kind of biological characteristic is obviously important in choosing your species. And then, of course, pharmacogenetics is a very important issue in biomedical research, and P450 genes vary significantly across primates. Now, I want to talk in more length about differences among populations within a species. And the most important difference is between Indian origin and Chinese origin Rhesus macaques. Rhesus macaques are the most widely distributed non-human primate alive today. They range from the east coast of China all the way across Vietnam, Myanmar, northern India, all the way up into, in fact, into Afghanistan. So it wouldn't surprise you if I told you that human populations from this part of the world and this part of the world were somewhat genetically different. It shouldn't surprise you at all that Rhesus monkeys are different. And in fact, there are a number of biomedically very significant genetic differences that drive differences in research outcomes. So one of the most important is the response to SIV infection, and I don't have time to go into the details, but Rhesus monkeys are the most important model in SIV research related to HIV vaccine development. Indian origin Rhesus monkeys respond to SIV infection different than Chinese animals, even though they're members of the same species. In addition, there are other differences, behavioral, neurobiological differences between Indian and Chinese Rhesus. The other species that's very important is the cynomolgus macaque, and this species is distributed here, ranges from the Philippines and through Borneo, Sumatra, Java, and into mainland Indochina. But it turns out that there's also another population of cynomolgus macaques. About 400 years ago, Portuguese sailors were moving around in Indonesia, and apparently they picked up a small number of cynomolgus macaques from somewhere in Indonesia and sailed across to a small island of Mauritius in the Indian Ocean and dumped a small number of animals in Mauritius. So there are now tens of thousands of Indonesian cynomolgus macaques that actually live on Mauritius, and they're feral, and they're extremely valuable for biomedical research because it was only a small number of animals that were transported. As a result, there was a severe genetic bottleneck. And rather than having dozens and dozens and dozens of MHC alleles like the native populations in Indonesia, the Mauritian cynomolgus macaques have a very limited MHC haplotype diversity. So if you're interested in the immune system, the genetics of the immune system is much simplified in the Mauritian cynos. So that's extremely valuable. But in fact, that has consequences so that Mauritian cynos respond to infection by Shigella virus differently than Indonesian cynos. Mauritian cynos developed sexual maturity at a different rate than Indonesian cynos. And if you're interested in pharmacogenetics, the gene expression in the liver of Mauritian cynomolgus macaques is significantly different than multi-locus gene expression in the liver of Philippine cynomolgus macaques. So this difference, although valuable for some kinds of research questions, has implications for different kinds of research reproducibility. Another example is baboons, and I won't go into a great deal a lot of detail in baboons, but there's multiple different populations of baboons, and they differ in their cardiovascular traits, they differ in body mass and ability to maintain body mass, and in other kinds of features. So there are a number of different levels at which population differences are important. Now, this diversity across populations is valuable when we can use it to pick the most appropriate animal for specific questions, as in the case of SIV research in Indian and Chinese rhesus. But it obviously has great potential for problems in reproducibility if we don't identify and characterize the origin of the animals. And now, finally, I want to finish with talking about the third level of variation, which is genetic variation within a single population. And of course, the most important population for us is not wild populations of macaques, but the research populations that we actually have access to. And so I want to thank various people at different primate centers for contributing samples. At the Baylor Genome Center, we have sequenced the complete genomes of 152 Rhesus macaques, producing deep whole genome sequence coverage for most of them and lighter coverage for the remainder. Most of these are Indian origin animals, but there's a few Chinese origin animals as well. When we looked at polymorphisms, single nucleotide polymorphisms in these animals, in the 144 Indian origin animals, we found 45 million SNPs. The 8 Chinese origin rhesus had an additional 6 million SNPs that were not found in the Indian origin animals. But by comparison, the 1,000 Genomes Project sequenced 1,000 people selected from all over the world to maximize ethnic diversity. In those 1,000 people, there were only 38 million SNPs. So we found more variation in 144 rhesus monkeys from colonies than is found in the entire human 1,000 Genomes Project. This is just another way of looking at that same information in the humans, whether you're looking at Europeans or Africans. Nucleotide diversity, which is a measure of kind of heterozygosity at the nucleotide level, is about 1 × 10⁻³ The Indian origin animals have variation about 2.5 times as much variation. Chinese origin animals about 3 times as much variation as you find in humans. And this is just a way of graphically representing that. I put this in for Dr. Begley because these are raw counts of SNPs, not percent fold change over humans. But this is the plot. If you plot the number of SNPs we find in a human population survey as we add individual humans to the data set, this is for Europeans, this is for Africans, and this is for Indian origin rhesus monkeys. So obviously, not only do they have much more variation, but the line's still going up as you get to 54 animals. So does this have biomedical implications? We found among our 51 million SNPs, we found 250,000 that are non-synonymous, predicted to change the amino acid sequence of the protein. In addition, we found more than 4,000 stop codons gained. So these are potentially knockout mutations naturally occurring in the NPRC population of rhesus macaques. Not every one of those is going to be a useful genetic model for human disease, but a very significant number of them are. We also found more than 3,000 splice site mutations. So collectively, these are going to be very important for implications in individual variation in the animals. It's not clear whether the macaques have a higher level of functional variation like this. We think they do, but we need more research to be sure. But clearly, the macaques have a lot of functionally significant variation. And here's just a few examples of the genes in which we found those de novo stop codon mutations and the human diseases for which they've already been associated, those genes. We also find functional mutations outside the coding sequence. So we looked at the ENCODE labeled transcription factor binding motifs and found more than 100,000 rhesus SNPs that are predicted to hit the motifs, and 28,000 that are predicted to reduce transcription factor binding, and 2,000 that are predicted to increase transcription factor binding. So clearly these have the potential to influence gene expression. So what can we conclude from this? First, rhesus macaques exhibit very high levels of genetic variation, both within populations and between populations. Cynomolgus macaques, I haven't shown you a lot of data, but they show exactly the same pattern. And in fact, this is typical for baboons and marmosets and all the other primates. It's not The primates that are unusual, humans, are depauperate in genetic variation, and we could talk about why that is if people are interested. So this variability within and between populations can, and I would argue already does, influence research results. So what are the opportunities going forward? The cost of DNA sequencing is coming down dramatically. It continues to drop dramatically. So we now have the opportunity to characterize genetic variation among individual animals within all of our research colonies. We could consider sequencing a very significant proportion of all the animals in the NPRC program. That would provide researchers with a remarkable database with which they could, in principle, select research subjects on the basis of genotypes for these functionally significant polymorphisms. And I would suggest that if that were done routinely, that would significantly improve reproducibility in non-human primate research. So, of course, as Roger alluded to, the technology continues to expand, and CRISPR and Cas9 system has been used in monkeys. So we are going to have targeted mutations in non-human primates. But remember that when you go to breed that animal that carries the mutation, you're not going to be able to breed it like the mice against an inbred background strain. We are still going to have to consider and take account of segregating variation in the population of animals we will use to expand and increase the number of animals carrying that targeted mutation. So CRISPR is obviously going to have a very important impact on our ability to do genetic research in primates, but it doesn't obviate the issue and the question about genetic variation. So what are the implications and maybe some recommendations for best practices? And with regard to reproducibility in research, I would suggest that researchers must evaluate the background and indeed the genetics of their study animals. That researchers should be choosing populations and choosing individuals that are appropriate to the research based on genetic composition. We now know how to identify individual mutations that are predicted to affect phenotypes. And how much time do I have left?
7.
Great. Okay, so I've been talking about sort of genetic variation from the point of view it's a problem, that it's noise and it's individual variation that is going to create problems. And that's, we have to think of it that way. But I also want to show you very quickly that genetic variation is actually very powerful in these systems, and we can make use of it once we define it and once we characterize it. And this is, I just want to very briefly, I don't have time to go into the details of it, but this is a collaboration that I have with Ned Kalin at the University of Wisconsin. And Ned has spent many years developing a Rhesus macaque model for risk factors for anxiety and depression. And what he does is he's developed a standardized test for juvenile Rhesus macaques that unveils individual variation in their behavioral response to a mildly stressful challenge. And the tails of that distribution of those Rhesus monkeys show behaviors which are very reminiscent of children who are extremely behaviorally inhibited and hence at much higher risk for anxiety and depression. So it's a Rhesus macaque, juvenile Rhesus macaque model for risk factors for anxiety and depression in humans. What we did was work with Ned where he had phenotyped hundreds of animals. It's about 250 animals, characterized the phenotype. In my lab, we genotyped those animals, or we sequenced the gene for corticotropin releasing hormone receptor 1, CRHR1. We found a particular SNP that is associated, yeah, that's okay, I understand. A particular SNP that is associated with behavioral variation in the animals, but it's also, the same SNP is also associated with metabolism in the anterior hippocampus tested using PET imaging. So the PET imaging results are taken while the animals, the PET imaging uptake of the radioligand occurs while the animals are undergoing the testing. So we can see whether, while they're responding to the mild stress, how active are different regions of their brain. So what we found is an individual polymorphism that influences the activity of a specific part of a neural circuit related to anxiety and fear-related behaviors, and that change in the neural circuit influences the outcome behavior. This is interesting to us for a number of reasons, but one of them is that SNP that's most important is right at the 3' end of exon 5. Probably affects the splicing across to exon 6 and exon 7, Exon 6 is actually a novel evolutionary innovation, so that macaques, chimpanzees, gorillas, and humans all share that protein sequence for exon 6. Lemurs and non-primate mammals just don't have the same form of the protein there. They really don't use exon 6. So we think that this splice site mutation may be altering the inclusion or exclusion of exon 6 from the final protein, and as a result, changing the function of this receptor. But I raise all this in the context of reproducibility because we first discovered this mutation and this association with behavior in the Wisconsin Primate Center, working with Ned Kalin. We have since worked with Judy Cameron, who's at University of Pittsburgh, but Judy measured anxiety-related behaviors and a whole range of traits in Rhesus macaques from the Oregon Primate Center. We genotyped the same SNP in the Oregon animals and found that, once again, the same SNP influences a variety of different behaviors related to anxiety and depression in these Rhesus monkeys. So that when we think about genetics, it's clearly creating individual variation among animals. We need to account for that and remove genetic variation when it's not the focus of our attention. But when we want to focus on genetics as a driver for individual variation, we can actually do reproducibility and replicable studies using that genetic information. So genetic differences can be considered noise when the focus of the attention is not on the genetics themselves. But genetic differences can actually be the signal that we're looking for, and that can, if we understand the genetics and how that influences phenotypes, that can increase reproducibility and replicability in primate work. So what near-term goals might we identify where we could benefit reproducibility in primate research? I think we ought to seriously consider doing a major effort to define the genetic composition of the most important research primates. By that I mean we need better whole genome assemblies and better genome annotations. But in addition, we need more information about genetic variation and gene expression across different species, different populations within those species, and in many cases across individuals. If we do that, if we develop a better characterization of the genetic variation within our research animals, that will allow us to choose animals on the basis of functional genetic variation. We can actually target research projects and select animals much more effectively so that researchers can actually choose animals that share the same functionally significant variation if they want to take that out of the equation, reducing the biological noise. Or in other cases, obviously we're going to want, like our study of anxiety and depression in the Rhesus, we're going to want to include animals of different genotypes so we can understand and do robust tests of the effects of that variation on our topic. With that, I will close and thank the people in my group who did the analysis of the whole genome sequences, other people at the Genome Center, thank collaborators, and of course collaborators up at Wisconsin for the work on Synos and Ned. And thank you for your attention.
Okay, our third speaker that, our third speaker this morning is Dr. Monte Westerfield, who is a professor of biology and a member of the Institute of Neuroscience at the University of Oregon. Welcome.
Thanks very much. Thanks for the opportunity to be here and speak to you. I'm especially looking forward to the discussion later. So I would, my task this morning, my charge was to talk about zebrafish research and reproducibility issues. And I was told specifically to focus on why zebrafish. We've heard quite a bit about these important mammalian models for human health and disease, and why in heaven's name would someone think about studying fish? So I'm actually going to concentrate on that and then talk about why zebrafish and provide some examples about their current use in biomedical research, hopefully to convince you that this is an important model system. And hence, if it's an important model system, then we need to consider issues of reproducibility. So the research started in the late 70s, early 1980s with developmental biologists because of this long-standing observation that all vertebrates have very similar early development. And this suggests that the underlying mechanisms are conserved all the way from fish and reptiles up to humans. And by studying these organisms that may be more accessible experimentally, we can find out what some of these fundamental mechanisms are. And with the advent of the genomes and genome sequencing, we made an even more remarkable discovery that essentially we all share the same set of genes with very few minor differences. And in some cases, the functions of these genes, even at very high levels, for example, the EYA1 gene, which is important for eye development in animals as diverse as fruit flies and humans, seem to be conserved. So again, by studying gene function in more experimentally accessible organisms, we can perhaps gain some fundamental understanding. So why zebrafish? Zebrafish have very rapid early development, as shown here. This is the first 24 hours of development of a zebrafish embryo, sitting on top of the uncleaved yolk cell. The eye, ear, body segments all form within 24 hours. And throughout this period, all the cells are visible, so one can actually use cellular and subcellular resolution for experimental studies. Then with the advent of transgenesis, it's now possible to mark cells with cell-specific promoters that drive markers, typically fluorescent markers, that are expressed in these living cells so that one can actually watch development, physiology, and so on in live animals. So here is an example of heart cells, endocardial cells, that are labeled with GFP transgene in the live animal. The resolution can be extremely great. That's a little hard to see. Individual cells, here's the pulsatile movement of blood cells as the heart is beating in a live animal. And perhaps most remarkably, it's possible now to monitor the activity of every neuron in the live brain. Here's the optic tectum, and this fish is going to experience a flash of light here shortly. You can actually see axons heading down the reticulospinal system telling this fish it better swim away quickly. Again, because of their small size and rapid development, it's possible to do very high throughput experiments. High throughput screening, for example, is illustrated in this system, where individual fish are plated into multi-well plates by robotics, and there they can be they could be mutant fish, or they could be treated with various potential pharmaceutical agents, and then via microfluidics directed into high-resolution optics, where they can be spun around in 3 dimensions. Every cell in the body can be visualized, and of course with various markers one can actually look at genes turning on and off, can monitor enzyme and protein activities in high throughput. So I hope I've convinced you that the real strengths here are the in vivo analyses at high resolution. After all, if you're going to do drug testing, doing it in cells, human cells in culture, for example, is certainly a reasonable approach, but doing it in the whole animal where you have the kidney, the liver, metabolism going on has obvious advantages. And most importantly, these analyses can be done very quickly and hence relatively inexpensively compared to some of the more expensive, particularly the mammalian models, as was just pointed out. So for these reasons, they've been adopted by a wide range of researchers, and I'd like to just sort of illustrate that in the next couple slides. So as I mentioned, zebrafish research started in the 1980s just with 3 laboratories, all at the University of Oregon. And there's been a steady and pretty dramatic rise in their use in biomedical research. Now there are thousands of researchers in over 40 countries that are using zebrafish. And that, of course, gladly has resulted in a consistent rise in research productivity, even up until today, in this time of austerity with reductions in the NIH budget, which have had pretty dramatic effects, as shown as an indicator of PubMed publications for humans, mice, and even fruit flies over the last year or two. And yet during that same period, we've seen very little impact at all, in fact, increasing productivity using zebrafish. So I think there are a variety of ways to interpret these data. But one interpretation might be that researchers that are using these more expensive models may be augmenting, or perhaps in some cases switching to the less expensive zebrafish model. Maybe there's some scientific reasons as well, but I think the correlation with this time period is kind of compelling. So what can you really do in terms of human health? Here's a list of NIH grants that were funded last year by using zebrafish models for human diseases. These are human disease terms. And to help your eyes a little bit, these include ones you might not expect: Alzheimer's disease, autism, obesity, schizophrenia, and even sleep disorders. So it isn't simply developmental biology anymore or basic physiology, but in fact a really broad range of studies can be done. Now obviously we're not going to study mammary gland development in zebrafish, so there are many areas that for which this model is going to be inappropriate. So I think we can say pretty safely that current biomedical research is very widespread and it's growing. So we need to think about this seriously as a model. It's still certainly not as broad spread as mouse work, but there are probably more researchers using this than are using monkeys at this point. So let's think a little bit about reproducibility, and I'd like to concentrate on genetic technologies, because that's by far and away the majority of research studies that are ongoing. The genetics started initially, and until today, the real strength of the system is classical forward Mendelian genetics. That is, you induce mutations randomly and then look for really interesting phenotypes in the particular system that you want to study as a means of blindly discovering gene function. And this really took off with the work of George Streisinger in the 1980s, making clonal lines and very high throughput methods for screening for mutations. And there are now many thousands of mutants that have been discovered this way. And then after the fact, one has to figure out what the gene was that was affected. So you do function first and then the gene later. But with the advent of the whole genome sequence, it's now, of course, possible to target genes, as we've seen already in Roger's talk. And the earliest technique that was used in zebrafish, and still the most broadly used technique, is to use morpholinos, which are antisense oligonucleotides that are targeted either to the translation start site or to splice blocker sites of the RNAs, mRNAs, and hence will disrupt expression of proteins and create a knockdown of the mutation, of the gene function. As I said, this is really widespread because it's very easy to do. One injects the one-cell fertilized eggs and then can screen directly in the injected animals for the knockdown. There are a number of problems that affect reproducibility that I think have really kind of plagued the field. The morpholinos themselves at high doses can be toxic, so you have variability in the response of the animals that are stressed by this injection. Because you're injecting once at the one-cell stage as the animal develops, the effectiveness gets diluted over time, so there's a time-dependent change in the effect. And the targeting it relies upon just binding of this antisense oligonucleotide to the RNAs, and hence any genomic polymorphisms can alter that binding. And hence, if you have polymorphism within your laboratory stock, or certainly between laboratories, this can produce different types of results. In particular, off-target effects, where the morpholinos are actually affecting protein expression from other genes. So it's always better to have a mutant, and with now the ability to do high-throughput resequencing, there's a centralized project, the zebrafish mutation project at the Sanger Institute to induce to screen for complete loss of function mutations in every protein coding gene in the genome. They're up to 45% of the genes have been knocked out. We anticipate by the end of next year or sometime in the following year to have knockouts of every gene in the genome. And because of the small and rapid techniques that are available, these animals are being phenotyped. So that means that we will have the first complete vertebrate phenome within a year or two. These mutations, of course, are randomly induced and are selected then on the basis of sequence to be loss of function mutations. But in terms of modeling human disease, we often need to have a much more precise genotype that matches individual patients. And CRISPRs have already been described this morning, so I won't go into this. But they allow very precise cuts that can produce indel mutations. It's also possible to use homologous recombination to insert sequences. And this is extremely successful in zebrafish. One can inject the animals and oftentimes study the injected animals directly because there's a relatively low level of mosaicism of these genotypes. So with this type of technology, it's now possible to do really very precise, to generate very precise models of human genotypes. And I'd like to just illustrate this with one example. So this is a family that has two daughters who have Usher syndrome, which is the leading cause of deaf-blindness in humans, combined deafness and blindness. And two of the daughters are homozygous for the most common gene that produces Usher syndrome, Usher2A. And the one daughter presents as a very typical Usher2 patient, but the sister has a much more severe phenotype, a very rapid retinal degeneration. So the thought was that there may be something else in the background here that may be modifying the action of this mutation. So our whole exome sequencing was done on the entire family, and it was discovered that this girl is heterozygous for a mutation in an unknown gene, PDZD7. So it's possible that this heterozygosity could be contributing to the more severe symptoms that she has. But as you can see, the evidence for this is extremely weak. She's heterozygous, and it's apparently a de novo mutation because neither parent is carrying that genetic variant. So you can't really tell the family much on the basis of this. So the obvious thing is to make an animal model with this genotype and compare this genotype to this genotype and see if, in fact, there is an enhancement of the symptoms. And so this is easily done in zebrafish. And what one sees here in blue is the amount of retinal degeneration that's occurring in the zebrafish, which has this same homozygous configuration as the two sisters. And then adding in a half dose of the PDZD7, which by itself has no appreciable phenotype, exacerbates the phenotype. So here the genotype matches the genotype here and the phenotype, and hence has been used to validate genetic interactions between PDZD7 and USH2A that then could be tested by in vitro binding assays and understanding how these proteins, whether these proteins really interact or not. And there's a physiological basis for this, which has now been done. And within about 6 months to 9 months of publication of this result, this gene was in the clinics. It was on the panels. So if you go in for genetic counseling for retinal degeneration, this gene will be included. So that shows how one can go very rapidly from the bench to the bedside using this sort of animal model. So I've sort of touched briefly on what the issues are. Several of them have been described already. In particular, the fact that we now have low-cost sequencing, rapid sequencing that should be available in essentially every laboratory. Because I mentioned the problems that we have with genetic variation among the individuals. We saw that that's true in all these systems. Clonal strains are available in zebrafish, and there are more that are being generated, but the vast majority of laboratories don't bother to use them. They're not as healthy, they're not as fecund as the outbred strains, and so those are typically the strains of choice. So one way around that is now, again, because of low-cost sequencing, it's possible to get complete maps of all SNPs. So you can actually genotype the individual animals that are being used in a particular experiment, the individual animal that's being used in an experiment. And some labs are starting to adopt this technology now so that you know for sure exactly what your CRISPR is doing, and hence can increase the reproducibility. And I mentioned that I think isogenic strains are extremely important too. These sorts of things are going to cost money, and also education of the research community that they're important, and it's worth spending the money. So with that, I will just summarize. I hope I've convinced you that zebrafish can be a good adjunct model for studies of human health and disease. And I'll leave you with the thought that we still need tools. Thank you.
Thank you, Dr. Westerfield. Now, if we can have all the speakers come up to the front table. And everybody has their questions ready. It's a very interesting session. A lot of hardcore science there, and a little bit different from the tone and focus of yesterday's session. So I hope you're ready to have a good discussion about this topic. So please go ahead.
Well, I thought all three lectures were great. I really appreciate the differences and the points you made. I'd like to add a couple of more variables that I think are important as we talk about the reproducibility. The first one is age, and it hasn't been mentioned. But in primates and in mice, the age at which you study them very much affects the outcomes, and I presume that's true for zebrafish. So I think we have to keep that in mind. The second is the use of the word models. I don't like the use of the word models because most of the time they aren't models, and I'm speaking from my own field. DB mice do not have any form of human diabetes, neither type 1 nor type 2. And I think we have to stop calling them by a human disease unless we know that it is the human disease. And I think that has been misleading, certainly in the field of obesity, OB, for example, and in the area of diabetes, and presumably that may be the case in Alzheimer's and some of the other diseases. And my third comment is on phenotyping. I don't believe in standardizing the phenotyping. I think that's a difficult problem because one standard may not be the right standard. But I do believe in detailing the phenotyping, and I think the phenotyping perhaps needs to be in an appendix of a paper or in something online. Phenotyping is critical. For example, glucose tolerance is not a single item. It is at least 15 different items. But if the phenotyping process is well defined, then you can make your own choice as to which.
Any comments from the panelists?
So could I just say on standardization, I actually hate the idea of standardization, but I think it is so impossible now to understand in terms of behavioral phenotyping. I'll just give that example. It's impossible to know what has been done. Putting 25 pages in a 125-page supplement doesn't seem to me a very effective way to get that across. And if there is an agreed upon standard for 3 assays or for the way to do one, or a centralized testing place that can take multiple models and do that, it might be a relatively painless way. I agree there should be nothing that seriously infringes on the ability of the investigator to determine what the best assays are.
So I'd like to respond to the issue with models. And I take your point that a lot of different things get lumped under the category of models. But from the point of view of non-human primates, I think in many, many cases, maybe models isn't the right word for primates because, in fact, they have exactly the same disease as humans.
We agree completely,
really,
which is why I object to the term model.
It's, you know, the physiology is so similar that we're modeling it because we're not studying it in a human. But when you study obesity or progression to AIDS or various other neurobiological processes in primates, you're actually studying the same disease. So maybe I'll take your point.
I agree with you.
I appreciate your point about reproducibility and phenotyping. And I think maybe there's, I see it as sort of 2 kinds of phenotyping. One that probably needs to be standardized and one that not so standardized. These high throughput methods, so the comp IMPC and the phenotyping that's being done for zebrafish, I think we really need to have standards because they're not all being done in the same laboratory, but they're all part of the same project. So there has to be some kind of agreement. But I also completely agree with Roger that in the laboratory, you need to do the experiments, right, that are going to give you the answers to the questions that you're asking. And I think it really constricts the freedom of thought and the ability to be creative if there are standards that one has to adhere to. So I think it's kind of apples and oranges, and we need to be clear about which apple or orange we're talking about.
Okay. Here, and if you could state your name and affiliation too when you ask a question, appreciate it. Thank you.
I'm Jim Marks from UPenn. Just to add another thing that may account for a lot of lack of reproducibility that we see in studies is that recently there's been a lot of work in the effect of the microbiome on the phenotype and how important that is in expressing the phenotypes. And certainly when you say, a C57 black 6 mouse is not the same as over here, that difference in the microbiome may really be accounting for a lot of differences that we haven't appreciated yet. I wonder if any of you could elaborate or have any experience with that kind of thing.
Yes, I couldn't agree more. So there have been really interesting experiments both in mice and in zebrafish showing how germ-free animals are very different. Obviously, there are problems with development of the digestive system, but even ex-germ-free animals, that is, animals that are then seeded with particular strains of bacteria, show very different phenotypes in particular assays. So it's yet another variable that needs to be thought about that the vast majority of researchers probably don't. And probably not every laboratory uses exactly the same feed for their animals. Very good point.
Back to this microphone, please.
Yeah, Michael Festing here. I think it's obvious that genetics is a very important variable that should always be controlled in experiments. In mice, Flint's group found that there's 5 times more variation in our outbred stocks of mice than there is in the human population. But there are many people who still don't use genetically defined animals. And one of the reasons was a point that Roger nearly made. The excuse for not using outbred animals is we wish to model humans. Humans are outbred. therefore we should use outbred laboratory animals. And people think that's a logical statement, but it isn't. Because if you look at a different character, we wish to model humans, humans weigh 70 kilograms, therefore we should use 70 kilogram animals, you see straight away we wouldn't be using rats and mice. So that is not a logical statement to justify the use of outbred animals. People should be thinking a little bit more carefully, and they should, all the people using rats, for example, really should be thinking about controlling genetic variation, which they don't do at present.
I'm all for being careful. I think, though, that ignoring genetic variation because you're using an inbred mouse is just as incorrect as ignoring it in using an outbred animal. So one has to think about what a genotype is, what that genotype is for, and how it's going to interact with the particular changes to that genotype that you're making. It probably, in many cases, will mean more variability, which means looking at more individuals and considering the variability of the phenotype as an outcome, not an annoyance that makes the statistics more difficult.
Can I just come back to you on that? You said something about the variability of skeletal measurements. I've got 2 papers in Nature on the variation in skeletal measurements, which shows quite clearly that the shape of the mandible in mice is exquisitely highly inherited, such that you can identify an individual, which strain an individual belongs to. And the variation in outbred stocks is greater than the variation in inbred strains.
Sure, I did not say that every measurement of the skull will be different. I said that there are a few measurements, precise measurements, that are different.
And I think another aspect of this is that ultimately we want to be able to understand human biology. We want to take our animal research and apply it to make predictions about what we would find in the human population. And to make robust predictions about what we would find in human studies based on inbred animals, you want genetic variation in your population, whether it's outbred mice or outbred primates. You need robust analyses at the animal level that are going to translate to the human situation.
Can I get back there? That is a fallacy, because if you have uncontrolled variation, you fail to detect an effect because the power of your experiment goes down. So you must control the variation in order to see an effect. Then you have to interpret whether that effect is important.
But if you control the variation in a way that doesn't occur in nature or in the population that has the condition that you're trying to model, then you're maybe advancing some kind of knowledge, but you aren't doing any good for understanding the disease mechanisms. I think I'm not suggesting we abandon the use of inbred strains. I think that the development of tools for looking at genetic variation, as Monte suggested, is a superb idea, and hopefully will be coming within the range of this. But I don't think that we should either ignore the fact that genetic structures have evolved for a reason. Diseases occur in species that generally have much more complex genetic structures than laboratory animals do. So that's my only point. I think there's a middle ground in those points that we should look for.
I'll come back to you privately.
Thank you. Here, please.
Stu Zola at Emory University, Yerkes National Primate Research Center. This is really for Roger, and coming back to you, what I hope you intended to be was a provocative first part of your talk. And I'm going to try and rephrase what you said, and that was that people trying to replicate are not the ones who got it to work in the first place. Can you kind of elaborate on that? Because if I were a journalist, for example, and heard you say that, I would say, well, then the world is lost. We're never going to get this right because, in principle, we are supposed to be putting out there in the scientific literature the ability to replicate. If you're suggesting that just because different people are doing it, it's not going to work, then so please.
Yeah, I guess the ability to, if you take the world record long jump, and then I go out and try and replicate it and I can't do it, and I say, aha, it must not be true. I think that's sort of the mindset behind a lot of the way replication or failure to replication gets reported in the popular press, when there really is such a thing as somebody who's better in the laboratory than another person. So that actually was the point I was trying to make.
So that's something then that we need to inform the general public about and Congress about as well. And I think it was you who said that if the student can't do this within a week, then you ought to get a new student or something of that nature. So there really is both an art, there's an art to the science, and that's something that's going to be important to make sure people understand more than they probably do. So, yeah.
Thank you. Here, please.
Hi, my name's Lynn Jackson. I'm a veterinarian trained in comparative medicine and toxicology. I currently have no affiliation, so if anyone has any open positions, I'd like to talk to them during the break. At any rate, I spent the last 17 years of my career in the pharmaceutical industry, and I'm very familiar with the concept, painfully so, faster, cheaper, better. And some of the comments that have been made in this morning's sessions, which I thought were all outstanding, get to points that were made, for example, by Dr. Rogers. Well, because this was a really expensive primate model, we spent a lot more time thinking carefully about experimental design. What concerns me, and we've talked a lot more so yesterday about incentives and disincentives for doing consistent, high-quality research, what I'm concerned about is that cost will be the only driver to choose animal models, and that might not be the best way to choose the right or the best animal model. So what can we do about that?
I think I agree completely. We need to select the animal model which gives us the best information and gives us the best ability to translate that information to the human situation. And there's always going to be a diversity of laboratory organisms that are useful for a whole variety of different purposes. But the translatability of the information to the ultimate goal must be, I guess, the primary consideration. And other issues like cost and things have to be second. It doesn't help to do an inexpensive experiment that doesn't give you the right answer, or the answer that you need.
We agree. Thank you.
Maybe I could just add oh, I'm sorry.
Sure.
Sorry.
No, go ahead,
I
please.
was just going to quickly add to that maybe in these cases where the correct scientific judgment is used to pick the animal to use, that the less expensive system could be the additional validation, right? It would help actually with reproducibility and help in terms of supporting particular conclusions if you can use a couple of different systems and come up with the same answer. So it could be an adjunct.
Yeah, I think that's well said.
Thank you.
Synergy between the models, and when you see robust findings that are true for multiple systems, then you have even higher levels of confidence. So yeah.
Thank you.
I have a question on the origin of the Rhesus monkeys that you mentioned. Anyone using monkeys, particularly in behavioral tasks, knows that certainly the largest source of variation is the variation between the individual monkeys. It's huge. So I was wondering, how do I tell whether I have an Indian monkey or a Chinese monkey, one? Two, do the breeding colonies pay any attention to this? And three, is there any way of testing, other than doing the genome, to see which type of monkey you have?
Okay. So this is really good because for 2 days we've been talking about really tough, hard problems that don't have direct solutions, and I have a very clean and direct solution for you. I'm the chairman of a working group for genetics and genomics for the National Primate Research Centers. That working group identified this problem, and we have now developed a SNP test, which is basically 96 SNPs that you can genotype for about $50 in any animal that you want to test. And that panel has the capacity to distinguish whether an animal is Indian origin, a rhesus monkey is Indian origin, Chinese origin, a hybrid of 50-50, and it can even tell 25-75 admixed animals. We can't go less than about 25%. But there are multiple labs that are now implementing this test. All the primate centers are using this kind of technology to confirm that they're the animals they think are Indian origin or Indian origin, to identify the animals that are suspect. So we can talk at the break, but there is a simple, direct solution to that problem.
I would like to make 2 comments. The first one is like, you talked about, scientists need to be careful about the genetics of the animal that they're using, but I would like you to comment on the fact that, in my opinion, the reproducibility is also, and the lack of reproducibility of certain data is also due to the fact, of like how the animals are being taken care of and like what is the environment of the animals in the labs. And I mean, there's this recent publication showing that mice are, reacting differently to male versus female caretakers. So I think this is like a very important thing. I mean, I've been in a lab where we're studying metabolism, liver metabolism, and one of the students was doing the experiment at night because he was from Thailand and was jet lagged, and the other student was doing the experiment during the day, and the data were totally the opposite. Nothing was reproducible. So I think there's like very, important points there. And the other thing is when you talk about making like the right choice on the animal models and the cost, I think it's important that scientists choose the model that is not just appropriate, but don't choose a model just because they know that this model will give them the data that they need in order to publish in, high impact journals. So I think those are very important too.
Well, maybe I can start about the environmental situation, because I think it's, well, I just don't know zebrafish as well. I'm sure that environmental factors influence behavior and physiology and growth and development in very important ways. I can't speak to that. But for primates, you're absolutely correct. The housing conditions, the social conditions, whether animals are pair housed or group housed, all have tremendous effects, not just on behavior, but on physiological parameters, on a whole variety of things. So absolutely. But the primate centers are focused on that, and are every one of the primate centers has a behavioral enrichment program. Every one of the primate centers pays attention to the psychological needs as well as the nutritional and housing needs of the animals. But you're absolutely right. It's a variable that does cause individual differences in experiments, but it's one that we try to deal with as best we can.
I think seasonal variation is another thing we don't take into account. Inbred mice that have lived hundreds of generations in a closed, light-controlled colony still go through very definable breeding depressions during the year, and we don't shower in our facilities, most people don't. So in the springtime, you're coming in with a lot of pollen. In the fall, you're coming in with a lot of leaf mold, and they're primarily olfactory creatures. So I think they know perfectly well what time of year it is. We also ran into that in an experiment looking at resistance to colon tumors in animals, and I, never having looked deeply into mouse food before, saw that there was a constant level of all the different nutrients, and I didn't realize that the percentage of different grains and fish oil and so forth that goes into that is recalculated every day to make the cheapest possible mix with those different things, and that tends to be seasonal. So when soybeans are being harvested, they're cheap, and phytoestrogens go way up in the food and so forth. So there are many aspects of that that probably have an impact on the microbiome, and maybe in other more direct ways on the animals as well.
I think we have time for one more question before the break.
So a couple of the comments were about the words we use and the way we talk about science. And I was struck, Roger, that you wrote, there is little reward and high cost of forming a hypothesis, trying to disprove it, and when you don't disprove it, to publish it. And it was to me, I'm reminding people I'm not a scientist, but all the talk yesterday about publishing negative results, I just wondered why you put it that way, and if you would, is that the sort of the prevailing attitude? I'm just trying to understand as a non-scientist why you said it that way.
Yeah, I'd hesitate to say prevailing, but if you look at the forces for those of us who work in a place where we essentially have a franchise, we pay 95% of our own salaries and fringe and 100% of everybody else and all the reagents and every pencil and all the chairs and everything. And then you go down to a grant success rate of 5 to 6%, usually at NICHD, which is also a 95% failure rate. It becomes really critical to get papers that have an impact, so they show up in the high-profile journals in a short period of time.
Yeah.
So getting a paper determines whether or not you get a grant, which determines whether or not you have a job.
Yeah.
So that's become a really disproportionate pressure, and the best source of that I can see is that the funding rates are way too low. So either we need more money or fewer scientists.
Right. That was yesterday's talk. Okay. Well, thanks. I'm taking notes for the session, too, so good clarity. Thanks.
Okay, well, I want to thank our speakers for incredibly informative talks, and thank the audience for some really great engagement. And thank you all for keeping us all on time. Appreciate that very much. So now we'll break until 10:45. Thank you.